everyone. It's my pleasure to introduce Stefan Eglen, the speaker today. Stefan received his PhD from Sussex in the area of computational neuroscience, examining the role of spontaneous neural activity upon development of nerve connections in the visual system. In his postdoctoral work, he mostly studied the development of the structure and function of the vertebrate retina. And now, since 2004, Stefan is part of the computational biology group of the development of applied mathematical and theoretical physics at the University of Cambridge. Today, Stefan will talk about theoretical models of the development of the nervous system.
Okay, thank you very much, Ariel, and thank you to Idan for arranging this seminar. My partner and I just arrived last Wednesday, when unfortunately all of the trouble started, and so it's been quite a stressful week, but I appreciate you all coming today. I'm sure you've got lots of other important things on your mind. So what I'm going to do this afternoon is to talk about three related problems in the area of neural development, and how I've been applying computational techniques to try and understand the principles underlying neuroplasticity, in particular the development of the visual system. So just as a reminder to get started, light enters the eye through the front end and gets transduced into electrical activity by the retina, shown inside and here, which we'll see in detail in the early part of the talk. The electrical activity is then passed into the higher part of the brain through the ganglion cells. The action potentials generated by the ganglion cells travel along the optic nerve into their primary destination in mammals, the lateral geniculate nucleus, and then that information is further passed forward to the visual cortex. So I've been interested for a long time in understanding the principles underlying the development of connectivity both within the retina and from the retina to its central visual targets. And the two main problems I'm going to highlight this afternoon are, firstly, a structural anatomical problem of how the neurons position themselves appropriately within a circuit, in particular the retina. And then I'm going to turn to the related question to say, well, once they've got their appropriate position in the circuit, how do they decide which other neurons to make connections to? Okay? So we've primarily used the vertebrate retina as our paradigm model for studying this, but our hope is that this will apply to other sensory systems, most notably the auditory pathway. So this is a picture of our local library here in Cambridge, and we effortlessly see this picture. A couple of things need to happen within the visual circuitry to allow us to parse this scene before anything else. First of all, we need a uniform sampling of the visual scene. We need receptors located across the entire visual world so that we can see the central parts of the scenes. And of course, if any part of the visual field doesn't have any receptors, then we end up with these blind spots. Okay? The second problem is then to get that information from the photoreceptors into the brain such that retinotopic information is preserved. So information from neighboring photoreceptors is kept and passed on to neighboring parts of central visual targets. And if we don't do that, we end up with scrambled visual scenes. Okay. So on to the first problem. How do neurons acquire their positioning? So what I'm going to do is to show you an example of what I mean. Paradigm example of that is population of retinal ganglion cells. So here the retina is taken out from the back of the eye, laid down, flattened. So the visual world would be projected down onto here. This is just one population of around 70 types of neurons in the retina. This population happens to be the class of alpha retinal ganglion cells in the cat. And what you can see is this exquisite patterning. Firstly, in terms of the cell bodies, the thicker there, and then the thin dendritic protrusions. We call these patterns retinal mosaics because of the way that the cell bodies and their dendrites tend to tile the retina. Okay, if you follow each dendrite out, they seem to come up and they only have a small amount of overlap with the dendrites of the neighboring retinal cells. Okay, so hence its name for a mosaic, just like a tiling property. We have looked at problems of the dendritic development of dendritic morphologies. What I'm going to focus on this afternoon, however, is simply the cell body positioning, so the central position here. So if you look at this, just considering the thick black blobs, as we take them as point processors, for example, we can see they cover the entire surface in what we call a semi-regular pattern. And I'll quantify what I mean by regular shortly. Okay. No two neurons tend to become too close to each other, nor are they too far apart from each other. And what I'm interested in is to work out what are the mechanisms driving the development of this retinal mosaic. How do these neurons know where to go in the retina to acquire their final position in space? So the approach has been to work with the experimentalists who've been cataloging these types of cataloging various types of retinal mosaic to try and come up with computer models that can try and account for similar patterns, both in terms of mechanistic models to try and model, for example, the development of the dendrites, and more in phenomenological models whereby we just try and come up with simple models that try and capture the spatial properties of these mosaics. What I'm going to do, with shortness of time, is just focus on the latter today, these more phenomenological models where we're trying to account for the pattern, with the hope that if we can account for the pattern, we may learn something about the mechanisms by which these are generated. So I'm going to introduce a very simple model called the exclusion zone model, also known by its dmin, the parameter for the exclusion zone. Or, alternatively, you've all hit this probably, and I understand it's a particular problem in Jerusalem, that of parking. This is the car parking problem. Okay? If you just have a stretch of road which isn't demarcated into individual car parking spaces, how many cars, if you're all arriving at different times, how many cars can you park along the street, and how close to maximal packing can you get? Okay? So this is the car parking problem in 2 dimensions. So we're going to start with a simulated, this is our simulated empty retina. What we're going to do is just add neurons one at a time, so cars are coming along. We're going to add neurons one at a time, pick a position at random, for example here. The center is the cell body, and surrounding it, the larger circle, is its exclusion zone. Okay. And the rule is very simple. I can park there, I can stay there, as long as nothing else is in my exclusion zone. This is the first neuron, so it's always accepted. Pick another place at random up here, and again, nothing is in the exclusion zone, it's accepted. This exclusion zone will vary slightly from cell to cell, as there's a bit of noise in here, and that size of the exclusion zone is what we refer to as the dmin parameter. What's the radius or diameter of this exclusion zone? So we keep going, and eventually here, for example, if we picked that as a trial position, we can't stop there because we've got a cell in our exclusion zone. So we go off and pick another point. So we repeat that, and then until we've eventually got an entire population of neurons in our field. Okay, so what? Okay. Now what we want to do is to compare these simulated patterns with observed patterns of retinal neurons to try and see if we can match what we observe experimentally. And over the last 10 or 15 years, we've been trying different statistical techniques to try and help us with this. I'm going to present 2 very simple metrics. There are lots of others around. So here's a population of neurons. What we're going to do to assess its regularity, in the first sense, is a very simple thing, termed here the regularity indexes. For each neuron, for example this one, we find its nearest neighbor, this one over here. We measure that distance. Take this one, measure its distance. So we've got a collection of distances. We then take the mean of those nearest neighbor distances and divide it by the standard deviation of those nearest neighbor distances. Okay. If this was a very regular population, we would expect the mean to be relatively high and the standard deviation to be relatively low. So the higher this number, the more regular the population, the mosaic is. Okay. At the other extreme, if you've got no regularity at all, just complete spatial randomness, the theoretical expectation for that regularity index is about 1.9. So we're going to be looking for values above 1.9. The larger the number, the more regular the population. That's a nice measure, simple, easy to understand. It's quite flawed, however. It's been compared to the problem, if you're looking at this, of measuring pairs of dancers, ballroom dancers, moving around in a square area. The nearest neighbor distance is always going to be very small. You've got a small distance to your partner. But it tells you nothing about the global layout of all of the dancers in the ballroom. So what we do is an alternative measure is for each cell is to put down a circle of a given radius and just to count the number of neurons that are within that radius, within that circle. So here we've now got not just one measure, we've got a function which is parameterized by the distance, the size, the radius of this exclusion of this circle. And so what we find typically is profiles like this red curve. The black line here is the expectation if there is no spatial regularity. Okay, so if it's under this line, it indicates regularity. I'll show you one example, I think, this afternoon of where it goes above this line, which indicates clustering. Cells are much closer, they like to be close to each other. This is called the L function. The mathematical definition is down there. So I can come back to that. The important thing is just whether it's below the line or above this line. So how well does it perform? The D-mean model, quite nicely. Okay, it's very simple. Got a 2-parameter model. It's simply the mean and the standard deviation of this exclusion zone parameter. So on the left column, we've got 2 different experimental data sets. In this case, a population of amacrine neurons that release dopamine. And in the bottom case, we've got the retinal ganglion cells, which are the off-beta class, which we'll hear more later. The circles are the individual somas. Surrounding each circle is what we've drawn as the Voronoi tessellation. That's the area of space which is closest to that cell. Okay, and the reason we draw that is partly just to help you see the regularity. You can see this looks quite random. There's a big variation in the size and shape of these polygons, whereas down here it's much more regular. And that's borne out by this regularity index. So these are mildly regular, and these are much more regular. So that's the experimental data. And on the right, I've shown you 2 examples of the D-mean model, the suitable exclusion zone parameters. Okay? And we get similar regularity indexes. Okay, well, what does that tell us from a biological perspective? It's not saying that the D-mean rule is actually what's going on in neurons. Okay? Clearly, it's not that cells are moving and coming down and getting rejected and going elsewhere. It's a much more dynamic process. And as I said, we've got more biophysically related models that capture that. But what it tells us is that the rules underlying this patterning can be entirely local. All you need to do is just to make sure that you've got a population of cells jostling for space, and they're just trying to push each other away and just say, don't come too close to me. You don't need any explicit rule to say, don't go too far away from me. That's a natural byproduct of just having a particular density of neurons in this population. Okay. But now what we can do is use this, albeit very simple, phenomenological model to actually look at more interesting questions. So now let's turn to another data set. So the retinal ganglion cells in most mammalian species, you'll find a pair of what we call complementary types. So for example, in cats, for the beta retinal ganglion cells, you can divide these neurons physiologically into what we call on-center and off-center responses. Whether they respond, ons I tend to draw in green and reds in off. So an on-center cell will increase its firing rate when you shine a light in its receptive field, whereas the off-center will increase its firing rate when you switch the light off. So this again is another mosaic taken from Heinz Vesely in the early 80s. And what I've done here is just reproduce it here, just drawing the cell body position with that cell type information, whether it's on-center or off-center. And when you look at this pattern, one thing that's jumped out to many people is that you seem to find that cells come in pairs. Okay, your eye is drawn to these pairs. They seem to come in opposite cell types. You don't tend to see too many cells of the same type too close to each other. Okay? Now, what does that tell us about on and off beta ganglion cells? Does it tell us anything about their developmental profile? Okay. So what we've been trying to work out, one of the issues is one of the sort of cell fate processes during development is when a neuron, how does a neuron, for example, this one, nose become an on-center beta ganglion cell, as opposed to an off-center beta ganglion cell, or as opposed to an alpha cell, or as opposed to some other cell. There's a lot of work underway trying to work out the cell lineage of different cell types in retina. Retina is a particularly nice example. It's much more complicated in cortex. So I think the retina is a good system for looking at this. So one of the reasons, striking reasons for why it might be that on and offs are actually not decided ahead of time, but actually come about through environmental interactions, is evidenced by looking at what happens across development with the dendritic trees. So now this is looking side on at the retina. The bottom of the eye is here, and the photoreceptors are up here. What you see is that at maturity, this would be an on-center cell. It has a very nice ramified dendritic tree in the what we call the inner plexiform, the lower part of the inner plexiform layer, and this one is an off-center cell in the outer part of the inner plexiform layer. So their dendrites tell you. You don't have to do the physiology. You can just look at the anatomy to tell you what kind of cell it is, whether it's on or off. Go earlier in development, and what you may find is that these cells don't look like that. They have quite broad dendritic trees. Okay. So one hypothesis that's been hypothesized, put forward, is that these cells don't know whether they're going to be on or off, but actually they're going to compete with each other, right? Such that they become different types. Okay, so if this one becomes an on, it's going to take up the local resources so that this one can't become an on and decides to become an off-center cell. Okay, that's just one hypothesis. Another hypothesis has been suggested is that of cell death, okay, which we'll look at. And finally, I just leave it open as some other combination. So let's look at this competitive hypothesis first. What I've done is to look at theoretical models, most prominently that of lateral inhibition of cell fate, and that's most normally expressed computationally with these the delta notch system, where the notch receptor is produced, the delta ligand is endogenously generated in each cell. It activates the corresponding notch receptor in its neighbors. So this one is generating delta and activating the notch receptors in the neighboring cells. The more notch receptor activation, the less delta that cell can produce. Okay, so the less delta that produces, the less it inhibits this one. So this one may generate more and more delta, inhibiting those two from having high delta. And by looking at the levels of delta and notch at maturity, you can then divide the population into two types. So what does this mean for our problem? So let's say that we've got an array of retinal ganglion cells. They don't know yet whether they're on-center or off-center, but they're going to undergo this spatial competition with each other, mediated through delta notch. So for example, these two are going to compete with each other, and simultaneously, these two are going to compete with each other, just so that they try and meet these constraints that the nearest neighbors are of opposite types. And that's what we see in stability over here. Greens are on-center, reds are off-center. I'll hold this thought for now as to whether that accounts for the patterns observed experimentally. Before we ask that, we'll look at an alternative model, that of cell death. This was proposed by Leo Chalupa quite a few years ago now, which just simply says that you may have some initial pattern of ons and offs, and all that you need to do to generate that nice mature pattern is just to get rid of the problem cells where the rules are broken. So nearest neighbors should be opposite type. So there you've got nearest neighbors which are the same type. Wouldn't it be really handy if cell death came along and just killed that cell, for example, so that nearest neighbors are no longer the same type? And when you look experimentally, Leo's group found that there was around 20% cell death in these retinal ganglion cells postnatally in the first 2 weeks in cats, and that suggested to them that cell death was a viable mechanism for this process. I've looked at that by building models of cell death to try and see whether these mature patterns can match the experimental patterns we observe. The answer is, unfortunately, neither of those two processes can account for the patterns we see. So what I'm plotting here is this regularity index, this nearest neighbor index, for the model results. This is the on cells and the off cells. The blue ones we don't need to worry about. For the model to recreate the experimental data, we need to get regularity indexes of around 5. So the error bars are here around 5. And we can see that lateral inhibition of cell fate doesn't work. Cell death doesn't work. The rescue that we thought about in the paper was to just put the two processes together, and lo and behold, if you have an initial undifferentiated population, which then differentiates through lateral inhibition, and then undergoes separately cell death, you magically get into that error bars. Okay, so that was our view circa 2002, and we then had a rethink a few years later, which was to say, well, hang on, maybe we're just being overly complicated here, trying to think about all these complex developmental mechanisms that are happening. Maybe that we've all been led aside by this nearest neighbor issue, and maybe there is no functional dependence between the on and the off cell arrays. Okay? Maybe it is that you just have a population of green cells that want to live in one space. You have a population of red cells which want to live in the same space, and they don't care where the greens and the reds. There's no interaction between the greens and the reds. Greens care about the position of other green cells, and red cells care about the position of other red cells. The only constraint that we introduced between the green and the red cells was simply that they can't sit on top of each other. Okay, you can't park your green car on top of a red car. So if we just allow some very minimal interaction between the two populations, just preventing the cell bodies from overlapping, can our exclusion zone models account for these populations, for these retinal ganglion cells? Okay, so my question to you is whether the pattern on the left looks like the pattern on the right. I hope you'll agree with me that it's a very hard thing to assess by eye, which is why we need these spatial statistics. So this is the map rotated 90 degrees from Heinz Fessy's group. This is an example from our simulation. The good news is that for all of the spatial statistics that we looked at, we find that the D-mean model accounts for the experimental data. So these are the L functions that I showed you before. The red curve as a function of distance is the experimental data. The black line surrounding it are the 95% confidence intervals. We can calculate p-values, but actually the easiest thing to do is a visual assessment, which is, does the experimental data lie within the error bar, the confidence intervals? In each case, for the on cells, for the off cells, for all of the cells, or for the cross correlation, we get a good fit. Likewise, this is the regularity index I've been talking about, nearest neighbor regularity index. This is the experimental data set. And these are 99 simulations from the D-mean model, and we get a pretty good fit for those. So from that, we concluded that there doesn't need to be any spatial interaction between these two populations. So when you see patterns like, when you see cells like this is just a visual illusion. You'll necessarily get them. Okay, our eye is drawn to them, but you just would expect them sometimes by chance with these two populations acting independently. Okay. So that's the problem of structural positioning, and I'll return at the end of the talk to seeing how this structural positioning has been looked at more recently. What I want to turn to now is a more functional question of how these cells wire up to each other. In particular, how do the retinal ganglion cell axons know which cells in the lateral nucleus to target? Okay. And in particular, I've been looking at the properties of sort of plasticity rules driven by the patterns of neuronal activity. Okay. And the key issue here is simply that the development of this circuitry happens before any visually evoked activity. Okay. So the photoreceptors are still developing. They're not functioning properly at the time that this circuit matures. So any visual activity, any sort of visual environment does not influence the activity of these neurons in the retina, unlike the ocular dominant story. However, even though the retina is not visually responsive, it's spontaneously active. I just wanted to show you some movies from a project that has been led by Evelyn Cerniger at Newcastle. Some of you may know Evelyn. I believe she did her PhD here many years ago. So Evelyn now has this very nice new multi-electrode array. It's got 4,096 electrodes. So each square here is reporting the activity of a single electrode. This is about 2.5 millimeters by 2.5 millimeters. This is the retina is placed ganglion cell layer down onto the array, and this is all recordings done in the dark, so it's just looking at the spontaneous activity. And what you can see are these propagating waves of activity. Okay. So neighboring neurons tend to be active at the same time. So even though there's no visually induced neuronal activity, the system generates its own spontaneous activity. Just on the right, Matthias at Edinburgh has put together this movie. It's just showing you what happens when you downsample this activity to a smaller 8x8 multi-electrode array, which has been the sort of the dominant system for measuring these waves. And you can see that the level of detail you get from these higher resolution arrays is quite significant. So let's build a model where what we want to try and do is try and use those patterns of activity to try and assess how they might influence the development of connections. So again, we've got our on and off retinal ganglion cell neurons, so the off and the ons. What we've drawn is a set of connections going into a single neuron in the lateral geniculate nucleus. So what we know is early on in development, the lateral geniculate neuron receives promiscuous inputs from many ganglion cells. And then during development, somehow some of those neurons, some of those connections strengthen and some of those weaken, with the idea that hopefully at adulthood, the neuron receives inputs only from neurons that are of the same type. For example, the on-center inputs. Okay? So this is a very well-studied problem, looking at plasticity rules in this context, for example. Through my PhD thesis was looking at covariance rule. Graduate student of mine, Juliana Georgieva, who's now with Chaim Sompolinsky in Harvard, looked at this for her thesis, where we took real recordings from mouse retinal ganglion cells at around postnatal day 10 to postnatal day 13. So what Rachel Wong's lab found was that these are the spike trains of 3 on-center neurons and 3 off-center neurons. Again, all entirely spontaneous activity. What you can see is you can tell the difference between the ons and the offs. The ons tend to fire with higher frequency bursts. They fire slightly earlier than neighboring off-center neurons, which fire with lower frequency and for longer. This is all spontaneous. So this is about, this is a recording, I think, from postnatal day 12, which is pretty much the earliest time you can really reliably get, from P10 onwards, you can get good identification of ons versus offs. Okay. What the field is missing is a good, reliable marker for knowing whether this is an on or an off cell. So you have to do the physiology to know whether it's an on cell or an off cell. Okay, but this is all spontaneous activity. So what Juliana did was just to look at building models where we take the real spike trains, put them into a model where the retinal ganglion cells are converging onto a single LGN neuron. We simulate the activity of the LGN neuron in an integrate and fire framework, and then we try and work out whether the correlations in activity between the retinal neurons and the geniculate neurons can drive the network such that it changes from this diverse state, receiving inputs from mixed types, to this adult state where the LGN neuron receives inputs from only, for example, the on-center neurons in red. Okay. And the first rule we looked at was the classics, or now classic, spike time dependent plasticity rule, where the relative timing of the pre- and postsynaptic spikes determines whether the strength of the connection should increase or decrease. And we've compared that with a more recent rule called burst timing dependent plasticity, proposed a few years ago by Dan Butts, working in the time in Carla Schatzer's lab, where, again, it's the relative timing pre- and postsynaptically, not of individual spikes, but of bursts. Okay, with the other difference being that classically the SDDP rule is asymmetric, so if pre fires before post, you get a potentiation, whereas conversely, if pre fires after post, you get depression of the synapse. Here, this rule is symmetric. So it doesn't matter whether pre or post fires, but first, just whether they're close enough together in time. Okay. The other issue being the time windows involved in spike time-dependent plasticity tend to be quite narrow, tend to be 50 plus or minus 50 milliseconds, whereas this rule goes out to plus or minus a second or two. I'm not going to show you the spike. Sorry.
What stage of development does this take place?
So this convergent probably happens throughout sort of the first 3 or 4 weeks in mouse. After birth. Yes. Sorry. So this is happening at these differential firing patterns peak at around P12, where this is still pretty much we think to be converging from multiple populations. Okay. The short answer is that SDDP doesn't work. You need to really put in a lot of extra constraints like normalization constraints. The big key problem being is just that the integration window is too short, which is one of the problems the burst time dependent plasticity has. Because really you need to look over the order of a second or so to see the difference between an on and an off-center cell. Okay. So here we're showing examples of the development of a set of connections. So we've got in each plot, we've got developmental time along the x-axis. The y-axis shows the strength of the synapse. For a group, in this case, 3 on and 3 offs. And in this case over here, for 5 on and 2 offs. Okay. And we find that burst time dependent plasticity reliably segregates the LGN neuron, reliably segregates the ons there. So you can tell the ons from the offs. In this case, all of the ons synapse onto the LGN neuron. Here, all of the offs segregate onto another LGN neuron. Yes?
The failure of SDDP is because of the temporal window or because of the shape?
So it's primarily, I think it's primarily the width of the window. You can get it to work under some circumstances, but it's very much data set dependent. So what you then have to do is, so we had about, say, 10 different data sets. So if it worked for one data set, it wouldn't then work for another. And we broadened the windows from 50 to 500 milliseconds, for example. Okay. So I don't believe we systematically looked at but then eventually the final thing is if you switch it from being asymmetric to symmetric, you're getting close to burst time dependent plasticity, which is what we were looking at.
So the asymmetry, essentially.
The asymmetry, I think, is important. Okay. So what we conclude is that during development, it may be that SDDP is a great rule in nicely tuned adult circuits where you've got fine, precise timings are really important. But this is development where things are relatively sloppy. Okay, and individual spikes can't be relied on, whereas bursts of action potentials can be pretty much more robust indicators of activity. Okay. And this fits in quite nicely with a lot of other work we did, for example, in other systems like ferrets. So this burst time dependent plasticity can account for the ferret system quite nicely. Okay, so now sorry. Yes.
How is it determined which type will
which dominate? It's partly to say what we've done is I didn't go into the details here. You can make some predictions based on the relative strength of the peaks. For example, the peaks of the cross correlation. The cross correlation curves between pairs of neurons, which we term the A parameter. So one thing is that the A parameter is instructive in saying if you've got a stronger correlation, it may be more dominant. Initial conditions are obviously another thing that pushes the decision whether it's become on or off. So for example, I don't know if you can see it here. These are 2 runs from the same system with the same real inputs kind of thing. The only difference being was just set the initial conditions slightly higher. So we've biased the off-center cells to be a little higher up to start with, and that's enough for them to run away. Okay, so what I want to do in the closing part of the talk is to then take us to what I call the final step. I very rarely get into the cortex. I spend most of my time in the retina, occasionally get into the LGN, or nowadays with mouse superior colliculus. But last summer I had the chance with a great graduate student, Victoria Hoare, who's now at Oxford, to look at a problem in the visual cortex. So how does this on and off information, so we've got on and off retinal ganglion cells effectively separately coding the visual image into the on pathway and the on pathway. At maturity, that on and off pathway is kept separate in the LGN, the primary target. But then we know that it gets integrated, on and off responses get integrated in the visual cortex. Okay? So what kind of interesting patterns can we get? Okay? So I'm hoping this cartoon will look familiar to most people, which is the classic Hublin-Wiesel type model of building an orientation selective cortical neuron. So we have got the visual plane represented here with a couple of retinal ganglion cells, the on-center ganglion cells here, and the off-ganglion cells represented here. So if this cortical neuron receives weighted input from these two oriented, slightly above, on-center, slightly above, and off-center, slightly below, what we've got is our classic bar detector. We get an oriented response from this cortical neuron. Okay, nothing new from that. This figure should also be familiar. This is the pattern of orientation preference of a group of neurons in V1. In this case, Mark Hubner's group recorded it using optical imaging, validating what Hublin-Wiesel proposed many years ago. So the color of each pixel tells you the preferred orientation of that collection of neurons to bars of different orientations. Okay. With the idea here that neighboring neurons tend to receive inputs that tend to have similar preferences to particular orientations, and what's highlighted here is what's called the classical pinwheels, whereas at the center, you've got the pinwheel center, surrounding it, you've got all of the orientations represented. Okay, nothing new there. There have been many, many models generated, proposed to account for these patterns. One of them that until recently was, I would say, relatively under discussed was that proposed in a PNAS paper by Bob Sudak in 1986, which is a very simple anatomical type model of how the retina might wire up to the cortex. So here I've got the visual plane of the retina with on cells and off cells. In this case, I've got the offs in red and the blues are the on cells. And that is expected to project in this model, is expected to project down onto primary visual cortex. So they somehow align to each other. So we were assuming that the problem of topography is already solved. So this neuron will project down to that corner, make connections to cortical neurons down in that corner. This neuron will make connections in this part of the cortex. Okay? So somehow that wiring problem is already, we assume that wiring problem has been solved. If that has been solved, what kind of properties do you get in the visual cortex neuron, in the visual cortex? So Dario Ringash has been looking at that in a few papers in the last few years, and it particularly got prominence last summer when they had a paper, we got the cover, for example, in Nature Neuroscience last summer, suggesting two key things. First of all, that the pattern, there's a particular pattern of the orientation pinwheels, which I'll show you, that hasn't been reported before. And secondly, that pattern is driven by the spatial distribution of the ons and off ganglion cells, these retinal mosaics that I talked about in part one of my talk. Okay, so when I saw this paper come out, I was particularly intrigued by it. So here's the first key finding from their paper. If you take these orientation pinwheel maps, so these things, and you perform an autocorrelation on them, in some particular, there's lots of technicalities and various ways of doing this, but the basic principle is that by looking at the autocorrelation of these pinwheel structures, what you find is not just the central blob, which you'll always get, the center of a autocorrelation, but you get these very dominant hexagonal patterns, secondary peaks in the orientation maps. Okay? And they've looked at this in a variety of maps from 4 different species. And the claim in their paper is that there's a regularity in the spacing of pinwheels in the cortex. And this has pretty much not been reported for with the pinwheels. Pinwheels have been counted and so forth. But this is a novel finding across these 4 species. Okay? So to try and account for that particular phenomena of of regular spacing in orientation pinwheels, they tried to take the earlier model that I proposed in Bob Sudak to try and account for these regular space pinwheels. So here's the model in essence. So you take a population of retinal ganglion cells, which are hexagonally spaced. So this is a hexagonal spacing of neurons with some given spacing. So this could be the on-center neurons. You take another population independently of off-center neurons with a similar spacing, so it may be D or it may be D plus or minus a little factor. Okay. And you've got a rotation angle theta. So you don't just overlay them straight, you can possibly just shift them one to the other. And when you do that, as you see here, you get these moire interference patterns. So I hope everybody can see that when you overlay them, you seem to get regular occurring moire fringe interference effects. Okay. So now, if you put these retinal ganglion cell distributions into the basic Sudak model, what you would expect is that whatever happens to the orientation selectivity of the neurons at the center of this fringe pattern here, you would get the same orientation at the next center because they've got the same relative spatial layout. So if you take a pair of neurons over here, you've got a red and a blue neuron. In this orientation, you would expect to see an orientation, a cortical neuron with that orientation. Okay, and that would be repeated wherever that pair is repeated. And sure enough, that's what you can see in the model, is that when you provide these inputs, shift relatively the 2 lattices, you get regularly repeating patterns in the orientation pinwheels. Okay? My concern with this was, first and foremost, that I hope that based on part 1 of the model, part 1 of my talk, is that retinal ganglion cells don't look like this. They're not on a regular hexagonal lattice. Okay, they're on this much looser prescription that they just avoid each other. Okay? So what Victoria did last summer was to compare how the orientation pinwheels form with a hexagonal mosaic, varying levels of noise, with our basic exclusion zone model. We've actually got one called PIP, it's the pairwise interaction point process model. So I showed you the cartoon I showed you before. They're all sort of getting up, moving. Sorry, they're being placed one by one. This is a much more dynamic one, where they're constantly moving around. Okay. And so we've compared the real patterns that we know of these retinal ganglion cell mosaics, according to various statistics, with these 2 different models. So here's some data. So the first column is a map taken from Gautier et al. 2009, this subset of off-center neurons. They look fairly regularly spaced. Okay, here we've got the autocorrelation of those points we see in an exclusion zone. I'll just highlight, for example, the L function we've seen before. Okay. So the hexagonal model proposed by the Paken-Ringash paper, either with low noise or high noise, generates patterns that are either far too regular compared to this, looks far too clumpy compared to this. And we can see, for example, the L function, it starts to diverge, the model starts to diverge from the simulations at longer distances. So low noise hexagonal mosaics are too regular. On the other hand, if we increase the noise, they become too noisy and they can't account for other features of these mosaics. Whereas our PIP model can nicely account for the autocorrelograms and the nearest neighbor distributions and the L functions. Okay, so what? My model is better than their model. You can. I would say that because this is the PIP model is what we, that we put in. That's not the important point. The important point is what happens when you put these mosaics into the model of orientation pinwheel formation. So what I'm going to show you is how the mosaics drive orientation pinwheels with either low noise, high noise, or with either low noise, high noise, or the pit model. So here, the first column is effectively the reproduction of Pake and Ringash. This is the orientation preference map. It either looks quite psychedelic, or some of you may agree that the pinwheels look quite regular, and that's confirmed in the autocorrelation plots down here, where you can get these regularly repeating spots. So far we agree with Pake and Ringash, is that if they're low noise hexagonal mosaics, you get regularity in the correlation. So you get regular spacing in the pinwheels. However, if you increase the levels of noise in the lattice to make it a bit more realistic, or using the pit model, what you find is that you no longer get these regular spaced pinwheels in the cortical orientation preference maps. So our conclusion to this, counter to what Pake and Ringash proposed, is that we don't believe the retinal mosaics have sufficient order to drive the regularity of pinwheels in the cortex. Okay, so that's my last data slide. I'm just going to conclude by wrapping up. So in the first part, I've shown you the neuronal spacing rules, the exclusion zone models. What we find primarily across a wide range of cell types is that these exclusion zones are fairly local. They don't tend to be more than, for example, 50 to 100 microns. Heterotypic interactions between cell types are rare. Okay, we don't tend to. We've got one example where we seem to see a positive interaction between cells of type A and cells of type B. Okay, so if anybody knows of any other cell type pairs where there are known to be positive interactions, I'd like to see them. Certainly in the retina, we only know of one strong case. Okay. Turning to part 2, I've shown you that spontaneous activity can be a reliable cue to driving the segregation of these information from the ons and off cells, and that during development, maybe the important time scale is not that of individual spikes and milliseconds, it's that of bursts and that of seconds, supporting Dan Butz's first time-dependent plasticity proposal. And finally, retinal mosaics, in my view, are highly unlikely to drive the formation of regular pinwheels in visual cortex. So I'd just like to conclude by thanking lots of collaborators from the various projects who were mentioned and the funding. Thank you very much.
There's time for questions. Yes.
Your very first neuronal density, it looks like when two centers were closer together than average, there was a dearth of dendrites between them. And when they were further apart than average, it looked like there was extra density. Is there a factor there?
So there may well be. Unfortunately, there isn't enough data to really. Sorry, let's just get back. Yeah. So this plot. All I can say is that we believe that the dendrites are crucial in the formation of the spacing patterns. So it's not as if cell bodies arrive, they do this thing in isolation, and then the dendrites grow. What we think is a lot more dynamic is that in the early stages, the dendrites do actually grow out and are actually one of the mechanisms by which this exclusion zone is enforced. So it may be that the further they've got to grow, the more the dendrites are allowed to proliferate, and conversely, the more sort of closer somewhere is, the less they are likely to grow. Okay, the evidence for that is kind of mostly indirect. The best evidence I've seen for that is where you put on drugs that inhibit microtubule formation. And when you do that, the dendrites start to collapse, and then the cell body mosaics also collapse. Then when the drug wears off a couple of days later, dendrites start to grow back, and the spacing returns. Okay, so it's very nice. That's pretty much the strongest evidence. But this is like gold dust, actually. This data was taken in 1981, right? 30 years ago, and it's pretty much the best there is. It's very hard to get very good, high, reliable identification of individual cell types. Andy Huberman's group is now one of them, for example, and also Josh Sains is putting GFP into cell type-specific things, retinal ganglion cell types. The big challenge there is, can you be really sure that that is an individual neuronal type? Okay. This is all single-cell injections, very painstakingly done, and the dendritic patterning confirmed that they were of one type.
About your model from the retina to the LGN. So in the context of other things in development, like solitary factors that affect the migration, the axon guidance factors, etc. So if I understand, a bit bigger picture is that the axons of both types of neurons will be attracted to the same region, and they will compete.
Yes.
So you will have still retinopathy across larger spaces, right?
Yes, we certainly expect retinopathy to be preserved. So I sort of jumped to the second stage of development, actually, by looking at the refinement of ons versus off. What we know in mouse, certainly, so that's about P10 to P12, is that the first week of development, that's when, for example, topography is really worked out. So by the time it gets to solving the segregation of different types of ons versus offs, it's already solved the retinopathy problem. And it may well be that during the retinopathy solving problem, those cell type specific firing patterns are not there. So they seem to appear at about the time they're needed to drive the segregation. Okay. So there's this sort of very step-like refinement. Topography, then, for example, the cell type segregation. Again, certainly there's a lot of evidence suggesting the role of molecular gradients in sorting out topography, and a less, I guess today, less so important role for neuronal activity, but still important for topography.
These first two questions really independent, separate, or they interrelated?
That of cellular spacing and that of wiring. At a first pass, I've always thought about them as separate processes, in the sense that putting out the cellular architecture is a prerequisite for then wiring them up, especially as that this spacing problem is done all within the context of the retina. So that can be done independently of what's going on with its axons, which are a long way away in the targets. I don't know of any evidence, major evidence, in terms of what's happening at the LGN for affecting the spacing in the retina, except you know that there are some competitive effects. So, for example, if you, because in most animals where you've got convergence of input from two eyes, if you remove one of the eyes, you've got less competition for the remaining eye in the target space. That may, in principle, affect what's going on with the spacing, because there may be less cell death. So they could be related. But I would say at a first pass, I expect the cellular spacing problem to be sorted out primarily before the second problem of the segregation of the axon terminals.
Any more questions? Okay, let's thank the speaker again.
Thank you.